Okay, so let's now proceed with the second part of our lecture, beginning with your ferritin. No? So if you can still remember the figure that I've shown you earlier, no, iron in the enterocyte no, can either be transported in the basolateral aspect, no, or they can be stored in enterocyte as a form of ferritin. No? So this is the major um, form of storage of iron in our body, in which we have around 1 gram of iron that we can actually store. Ferritin contains 24 subunits, in which the heavy chain contains your ferroxidase activity, and the light subunit or light portion is required for its stability. So in our body, we have around 50 to 200 micrograms per deciliter of your ferritin, and this is said to be an indicator of your body iron store. So definitely you can use ferritin in diagnosing iron deficiency anemia. Okay, we have such term here known as your hemosiderin, which are partly degraded form of ferritin that contains iron. This can actually be detected by different or by histological stains. If you can still remember, if you have transferrin saturation of more than 50%, that might be indicative of iron overload, in which your hemosiderin can also be expected to be seen. Okay, so this slide just shows us the regulation of your ferritin and your transferrin mRNA. So the rate of synthesis of your transferrin receptor as well as your ferritin is said or are reciprocally linked to the level of intracellular iron. Okay, so ferritin, as you can see here, is located in the 5' region of your RNA, while your transferrin receptor is located at the 3' end. Okay, so let's start first with ferritin. With high intracellular concentration of iron, no, if you can still remember a while ago, the what do you call this? The transport of iron in the cell. When you have high levels of iron inside the cell, the iron regulatory protein, no, which is seen here, will not bind to the iron response elements that is seen in the 5' region. If that IRP will not bind to IRE, that would lead to translation of your ferritin. Is that beneficial? Yes, because as you can remember, ferritin is the storage form of iron. If you have high levels of iron, you want to make a protein that would actually store it. Let's go to the next here. If you have low intracellular iron, IRP, no, or your iron regulatory protein, can now bind to your iron response element. When that happens, no, it would block the translation of the protein, so no ferritin can be formed. Okay, so that is beneficial because if you don't have any iron to store, why would you produce ferritin? So that is beneficial. Next, in your transferrin receptor, if you have high intracellular iron, no, that would again block the binding of your IRP to your IRE in the 3' region, no, in this case. No? So if that happens, this mRNA would be degraded and no transferrin receptor is then produced. Is that beneficial? Yes, because when you already have high intracellular of iron, would you still like to make the transferrin receptor to scavenge more iron? Of course not. So in elevated or increased intracellular iron, no, would result in the decreased production of your transferrin receptor. Okay. Next, if you have low intracellular iron here, of course, your IRP is now capable of binding into your IRE in the 3' region, and that would lead to translation of your transferrin receptor. Would that be beneficial? Yes. No, because when you have low intracellular iron, what would you like to do is to increase the formation of the receptors to scavenge more iron going inside the cell. So this process is beneficial. Again, the synthesis of your transferrin receptor and your ferritin are inversely related. Next would be your hepcidin. No? Your hepcidin is another plasma protein that is very important in controlling your iron hemostasis. So, what are the functions of your hepcidin? It decreases the iron absorption in the enterocytes. No, it also prevents the recycling of iron from the macrophages as well as reducing your placental transfer of iron. How does it do that? Paano ba niya ginagawa yon? So if you can see here, on the left would be your enterocyte, no, and on the right would be your macrophages. We've already encountered this, no, in our earlier discussion. So what does your hepcidin do? So you have prohepcidin that would eventually be converted into your hepcidin. So this would cause the um, what do I call this, internalization and degradation of your ferroportin. If you can remember the function of your ferroportin, that is the transporter of iron from the intestine, no, and the enterocyte going to the basolateral aspect. And on the macrophages, it's the one that releases your iron in the circulation. So if it would cause internalization of your ferroportin. Would there be a transporter to to transport iron out of the enterocyte and out of the macrophages? No. So if there's no transporter, can iron go out of the cell? No. So would there be iron that can be utilized by the cell that to produce heme? No. So that being said, it would lead to a decreased level of iron, no, and um, would also lead to eventually decrease in the synthesis of heme. Okay. Another thing to remember is that your hepcidin is upregulated, no, when you have chronic inflammation, no, and this is said to be the one responsible for the anemia in patients with chronic diseases, no, if they have malignancy, no, or other chronic diseases, no, your upregulation of hepcidin is responsible for the anemia there. Okay, so let's now proceed now with hemostasis and thrombosis. So just an overview, hemostasis is related to the cessation of bleeding from, take note, from severed vessel, no, from damaged vessels, whether it be it's an internal or an external cause of the damage. So hemostasis would have an initial period of vasoconstriction. No, this would actually try to limit the blood flow to the area so it can conserve the blood. Thrombosis, on the other hand, occurs when the endothelial lining, no, which is the innermost layer of your blood vessel, is damaged or removed. Okay, so both your hemostasis and thrombosis share three phases. That would be your platelet aggregation, no? That is also known as your primary hemostatic plug. You also have your formation of fibrin mesh, no, to actually stabilize this platelet aggregation or initial hemostatic plug. And once the body is done repairing this damaged vessel, there would be dissolution of the hemostatic plug or thrombus. Okay, so just an overview, your platelet aggregation, no, is stimulated by what do you call this, release of collagen from the injured vessel. This would also release your thromboxane A2 as well as your adenosine diphosphate, which is seen in activated platelet. Okay, this different signaling molecules, thromboxane A2 and adenosine diphosphate, would also activate other platelets, causing a change in shape, no, leading now to your formation of your platelet aggregation. Okay, So let's go first with your platelet aggregation. So your platelet adhere to the collagen via different receptors. No, you have your glycoprotein 1A, 2A, glycoprotein 6, glycoprotein 1B, 9, as well as 5, which is mediated by your von Willebrand factor. Okay, so platelets that are bound to collagen would change shape and release different contents of their storage granules. No, so among this, no, thrombin, which is actually produced in the coagulation pathway, is the most potent stimulation of granule release. Example of granules from platelet would be your alpha and your dense granules. No, what are the molecules that is seen in the alpha granule? Examples would be your factor 5, factor 13, as well as your fibrinogen. In the dense granules, you have your serotonin, no, which is a potent vasoconstrictor, adenosine diphosphate, which is needed for activation of platelet, no, as well as your calcium needed for the coagulation cascade. No? So an important or an acronym that I keep in mind, no, when to remember your dense granule would be your SAC. No? So your dense granule is actually a SAC. You have your serotonin, S for serotonin, A for ADP, and then C for calcium. Okay, so this slide may be overwhelming to you, but I would not require you to memorize all of this. What is important is to know. So how are you going to know this slide? No, so you just need to remember the molecule here, no, present on the outside, no. What are the receptors that they activate, and what are their second messengers? Okay, so thrombin, which is said to be the most potent activator of your platelet, binds to your to different receptor. Now you have your PAR1, you have PAR4, as well as your glycoprotein 1B, 9, and 5. So in turn, they would activate your phospholipase C beta, and this phospholipase C would eventually hydrolyze, no, your phosphatidylinositol phosphate. No, your phosphatidylinositol phosphate, once degraded, would be converted into your diacylglycerol and inositol triphosphate. This inositol triphosphate would then facilitate the increase in the intracellular calcium, increasing now your platelet responses. Your diacylglycerol now would activate another second messenger, which is your protein kinase C, that is also needed for the activation of your platelets. No? Thromboxane A2 would also do the same thing. No? It would activate the platelet, no, via the arachidonic acid pathway. Your ADP or adenosine diphosphate, no? So it's an example of purine, no? So it would activate purinoreceptor, indicated here as your P2Y1 and P2Y12. So once activated, no, they can um, actually also stimulate, no, your platelet responses. Prostacyclin, no, on the other hand, is a negative regulator of your platelet aggregation. So they inhibit platelet aggregation. That is secondary to increasing the levels of your adenylyl cyclase. Adenylyl cyclase would increase the level of your cyclic AMP, and as you can see here, it negatively affects your platelet responses. Your ADP as well, no, can also inhibit your adenylyl cyclase, no, producing also your platelet aggregation. Okay, so let's now go to your coagulation pathway. So there are two pathway that leads to formation of clot. Now you have your extrinsic pathway, which initiates fibrin clot formation in response to injury in the vessel, or intrinsic pathway that is activated by negatively charged surfaces in vitro, no, or in vivo as well. So some of the molecules that can activate your coagulation pathway, negatively charged molecules, includes your kaolin, no? And inside our body, no, damaged DNA that contains phosphate, no, can also activate your intrinsic pathway. Okay, so both this coagulation pathway has a common goal, no? So they want to activate your prothrombin to form your thrombin. Because thrombin is the one that would um, activate, no, or stabilize your clot formation. This thrombin is also needed, no, for conversion of your fibrinogen to fibrin. So what are the function of the different proteins involved in blood coagulation? Some of them can act as zymogen of serine proteases. So some of them are and has enzymatic activity. Your factor 12, no, is capable of binding to negatively charged surfaces, no, activating your intrinsic pathway. You have your factor 11 that is activated by factor 12A. You have your factor 9 activated by factor 12A and 7A. You have your factor 7, no, that is activated by 7A, 10A, and then thrombin. Your factor 10, no, which is the one that connects both the intrinsic and extrinsic pathway. So they are activated on the surface of activated platelets, either by the intrinsic 10-ACE complex. No, later on we'll be discussing it. Intrinsic 10-ACE complex consists of your calcium factor 7A and factor 9A, or the extrinsic 10-ACE complex composed by your calcium, tissue factor, and your 7A. Your prothrombin, which is also your factor 2, is activated on the surface of platelets by the prothrombinase complex comprising of your calcium factor 7A and factor 10 to form your thrombin, no? Okay, your thrombin, no, um like your other carboxyglutamate containing clotting factor is activated by your vitamin K. Next would be the cofactors. No? So they don't have enzymatic activity, but they are needed for the activation of the enzyme. You have your factor 8 no, that is activated by thrombin. Okay, your factor 5, which is also activated by thrombin. And it is a cofactor in the activation of your prothrombin by the factor 10A. So for you to activate prothrombin by factor 10A, you would need factor 5. Tissue factor, no, or factor 3 is a glycoprotein in the subendothelium, no, that act as a cofactor for 7A, no, in the extrinsic pathway. Sorry. Of course, you have your fibrinogen, factor 1, which is cleaved by thrombin to form your fibrin clot. You also have your thiol-dependent transglutaminase enzyme, no, your factor 13, otherwise known as your fibrin stabilizing factor. No, you have other regulatory proteins. No, most of them are anticoagulant. No, you have your protein C that is activated to activated protein C by thrombin. No, and degrades factors 8A and 5A. Protein S, which act as a cofactor for protein C, no, contains carboxyglutamate residue. So your protein S and protein C are actually anticoagulant that is also activated by your or dependent from vitamin K. And thrombomodulin, which is a protein on the surface of endothelial cells, binds to thrombin, which also then activates your protein C. So all of them are anticoagulant proteins. Okay, let's now first proceed with the in extrinsic pathway. What again is the um, what again stimulates the activation of your extrinsic pathway? That would be a damage to your vessel. Okay. Oops. Okay. So tissue factor, no, which is the first clotting factor that is activated here, no, or that would be exposed, no, um, in the blood vessel, they are actually located in the subendothelium of the blood vessel. So they are released no when there is tissue injury. Now tissue factor no would then interact and activate factor 7 to produce 7A, no, which is a vitamin K dependent clotting factor, which serves as high affinity calcium binding site. Now, this um, these factors would then what I call this activate no your clotting factor 10As no via your extrinsic 10As complex. So your extrinsic 10As complex is composed of your calcium no that is produced or that is needed uh, to generate clot no tissue factor that is released at the site of injury and clotting factor 7A. Okay. So how do they activate clotting factor 10A? So they activate clotting factor 10A by hydrolysis of arginine isoleucine bond no, in the protein. Okay, so once this bond is cleaved, no, it would lead to the activation of clotting factor 10. Okay. Aside from that, as you can see here in the figure, your tissue factor together with your clotting factor 7A can actually activate your clotting factor 9A. And this is an important concept because your extrinsic pathway is said to also activate your intrinsic pathway. Okay, so there. So tissue factor 7A activating your 9A. Your extrinsic 10 is complex, cleaving your arginine isoleucine bond, no, activating it to 10A. So we have already mentioned or we already touched about the different vitamin K clotting factors. So vitamin K is an important molecule in the carboxylation of the different gamma glutamyl or gamma glutamyl residue in the different clotting factor. So how does that happen? So precursors of different clotting factor, your 2, 7, 9, and 10. No? So these are the vitamin-dependent clotting factors. They need to be carboxylated no, or gamma carboxylated utilizing an enzyme, vitamin K dependent carboxylase enzyme. So what does the vitamin K dependent carboxylase enzyme do? So it creates an additional carboxylate group. Now, as you can see here, now in the precursor, there's only one carboxylate group, that's COO negative. No, but when it's activated by this enzyme, no, there would be two carboxylate group now. So once you form the gamma carboxylate group, no, utilizing your vitamin K as well as your carbon dioxide and oxygen, that would convert now the molecule, as you can see here, into a negatively charged molecule. Your carboxylate ion is negatively charged. That would then now serve as binding site for calcium. So calcium is positively charged molecule. So it would associate itself or bond with the carboxylate ion from the vitamin K dependent clotting factors or activated clotting factors. No? And it would also associate in the membrane phospholipids. If you can still remember, phospholipids are negatively charged molecule as well. So this will facilitate the bridging no, of your mature clotting factor in the membrane phospholipids of platelets where you need to form your clot. Okay? So parang papatungan niya yung activated platelets ng fibrin polymers para magkaroon ng clot. Okay? So there is actually an inhibitor of your intrinsic pathway that is called your tissue factor pathway inhibitor or your TFPI. So they are associated with lipoprotein that serves different function to inhibit the extrinsic pathway. So one would be the inhibition. It can directly inhibits your clotting factor 10A, no? And once it associates with clotting factor 10A, so they would form a complex, this can then inhibit your tissue factor and clotting factor 7A complex, inhibiting your extrinsic pathway. Okay? Let's now go to your intrinsic pathway. So sa intrinsic pathway, medyo mas madaming clotting factor. Okay? So you have your factor 12, factor 11, factor 9A, which is a serine protease, clotting factor 8A, which is a cofactor that we discussed a while ago. You also have factor 10 that I mentioned, serves as the bridge for the intrinsic and extrinsic pathway. You have your prekallikrein, no? PK is prekallikrein. You have your HK or high molecular weight kininogen. You also have calcium as well as your phospholipids. Okay. So again, what activates your intrinsic pathway would be negatively charged surfaces, no? So it can either be in vitro, no? Common yan, yung mga kaolin, no? And inside our body, we have negatively charged molecules like our DNA, which contains polyphosphate that are released, no? During cell damage. Pag na-damage yung cell, syempre lalabas yung DNA niya, including doon would be the phosphate groups that can activate the intrinsic pathway. Okay, so your factor 12A, 11, so factor 12, factor 11, high molecular weight kininogen and prekallikrein as well as the negatively charged surface can then proteolyze your kallikrein. Kallikrein no, is a product of your prekallikrein. So nanggaling siya dito. This can turn activates your clotting factor 12 to form your 12A. Clotting factor 12A can then activate more high molecular weight kininogen and more prekallikrein to generate ano uli ang product ng prekallikrein? That would be your kallikrein as well as your bradykinin. Now, ano uling gagawin ng prekallikrein? That can activate more clotting factor 12A. So in that sense, this is actually a positive feedback mechanism wherein the formation of kallikrein would actually generate more kallikrein to produce activated clotting factor 12A. So ganun lang yung ano niya, yung process niya. It would also activate yung 12A would activate your clotting factor 11 to 11A. Then your 11A would activate your 9 to 9A, no? So leading now to the formation. So as I mentioned a while ago, your clotting factor 8 is actually a cofactor. So this activation of the series of clotting factor would lead to the generation of your intrinsic 10-ACE complex. What are the component of the intrinsic 10-ACE complex? That would be calcium, which is needed always for clotting. No, you have your factor 8A here, which is a glycoprotein precursor that is a cofactor. No, which as you can see here is activated by your thrombin. You also have your clotting factor 9A, no, that when they associate with each other, no, they would activate your clotting factor 10A. And also with the intrinsic 10As, or extrinsic 10As, the intrinsic 10As complex can then cleave the arginine isoleucine bond now in your clotting factor 10 to convert it into its active form. So next would be the conversion of your or activation of your prothrombin to thrombin, no? So activation of prothrombin to thrombin requires the association of your prothrombinase complex, no? So prothrombinase complex is composed of your clotting factor 5A, no, as well as your 10A, no? Kaya natin gustong i-activate yung 10A because it's needed for the conversion of your prothrombin to thrombin. And your prothrombinase complex also needs calcium. Okay? So prothrombin is a single chain glycoprotein that is synthesized in the liver. It's also a vitamin K dependent clotting factor. No, it's otherwise known as your factor 2. Okay, that's why it has a uh, carboxyglutamate residue. No, you actually have 10 carboxyglutamate residue in your prothrombin. Okay, once activated, no, thrombin can actually activate your platelets. No, as if you can remember in the figure a while ago. And what is more important here would be the activation of your 13 to 13A. No, because this is your fibrin stabilizing factor that would stabilize the formation of your fibrin polymer. So next, no, tapos na tayo dun sa platelet aggregation, na-discuss na natin yung activation of the intrinsic and extrinsic pathway. Now we are ready to form your fibrin. Di ba yung next phase is formation of your fibrin mesh, no? So this conversion of your fibrinogen to fibrin is catalyzed by your thrombin. Okay, so as I mentioned, thrombin is actually an enzyme produced by the prothrombinase complex that has a potent stimulatory effect on platelet as well as converting other clotting factors. As you can see here, no, thrombin can activate your clotting factor 8. It can also activate your clotting factor 5 as well as your 13. It can also convert your fibrinogen to fibrin. So madaming action si thrombin. Okay, so I apologize for the interruption. I experienced a little bit of technical difficulties, so let's now proceed. Okay, so in the succeeding slides, we're going to discuss how thrombin activates your fibrinogen. So this molecule is actually an example of your fibrinogen. Fibrinogen or clotting factor 1 is a soluble plasma glycoprotein. No? So meron din siyang sugar molecule attached to it. So it has three non-identical pairs of polypeptide chain. No? So it has alpha, beta, and gamma. Okay? So as you can see here, they are linked by your disulfide bonds. So yun ang nagko-connect sa kanila. So they have disulfide bonds. And the amino terminal, as you can see here, faces each other while the carboxyl terminal faces away from each other. Okay? If you can see here in the alpha or in the A and B region, you have this green and blue marks, otherwise known as your fibrinopeptides. No? So this molecule, this fibrinogen, contains a lot of aspartic and glutamic acid. No? And in that sense, it has a negative net charge of negative. Okay, so that negative charge of your fibrinogen contributes to the solubility of your fibrinogen in the plasma. Okay? So, and because of that, no, dahil madami siyang arginine and glutamic acid, which are negatively charged in the circulation, they would tend to repel each other. So, yung isang molecule ng fibrinogen would repel another molecule of fibrinogen because of that charge, because of that similarity in charges. So it would prevent aggregation in the circulation because of electrostatic repulsion. Okay, so here, next. So, the serine protease thrombin, no? Ito na. Paano kino-convert ni thrombin? si fibrinogen to fibrin. So the serine protease thrombin form, no, would hydrolyze four arginine and glycine bonds in the N-terminal. Di ba yung nasa gitna, nandun yung amino terminal or N-terminal between the alpha and beta chain of your um, fibrinogen, liberating now your fibrinopeptides. As you can see here, no, maliliberate yung fibrinopeptides A and B. No? Once there's a removal of fibrinopeptide, ano napansin dito? Nawala yung larger portion and it would now expose a binding site at the E domain, no? Yung gitna, that is what you call the E domain. On the side, you have your D domain. So there would be exposure of the binding sites, no, on the E domain that would specifically interact, no, with the D domain of another fibrin. So kapag na-incorporate yung another fibrin, they would form bond, no, with the exposed binding site at the E domain. So that would produce now your fibrin polymer. So this fibrin polymer is said to be insoluble but is still unstable. No? So magkakadikit na sila pero yung bond nila is a non-covalent bond. So it's not that stable. So ano pa yung isang action? So ito na yung isang action ni what do you call this? Ni prothrombin. So it would cleave the arginine glycine residue, no, here para magkaroon ng fibrin polymer so that there would be binding of the E and D domain. So aside from that, no, your fibrin or your thrombin, sorry, would also activate your clotting factor 13, which is your transglutaminase enzyme, otherwise known as your fibrin stabilizing factor. Okay? So this fibrin stabilizing factor would then form bond, no? Magkakaroon siya ng bonding, as you can see here, the black portion, between the lysyl residue of one fibrin molecule and the glutamyl residue of another fibrin molecule. So there would be cross-linking between the two. So this now is a more stable, no, fibrin molecule. Okay? So not only that your thrombin converts your fibrinogen to fibrin, it also stabilizes it by activating your fibrin stabilizing factor. Okay? So how do we control thrombin? No, we have feedback mechanism, but what is more important would be the inactivation by your antithrombin. So antithrombin is the most important thrombin inhibitor. No, in fact, it can also inhibit other clotting factors like your 9A, 10A, 11A, 12A, as well as your 7A complex with your tissue factor. So, this is just to show you that your antithrombin, no, can actually inhibit the thrombin. No? But the activity of your antithrombin is greatly potentiated by binding with heparin. So heparin, mechanism of action is to potentiate the action of antithrombin. In that sense, now, your heparin would serve as an anticoagulant. So it would inhibit coagulation because it would potentiate the activity of your antithrombin. So when you don't have antithrombin, no, of course, the action of your thrombin is inhibited, no? So lahat ng mga function naman ni thrombin we have already discussed earlier. Protamine sulfate is actually a negatively charged molecule that can bind your heparin. So kapag nag-bind sa kay heparin, yung heparin pa paano magbabind kay antithrombin to potentiate its effect? Wala. So in that sense, now protamine sulfate can actually be used in cases of heparin toxicity. So this is the regulatory mechanism of thrombin. No? You have your thrombin associating with thrombomodulin. When they form complex, it would activate your protein C and protein S. Okay, so Protein C and protein S, as I mentioned a while ago, are anticoagulant. No? Anticoagulant. When this anticoagulant is activated, they would degrade clotting factor 5 and clotting factor 8A, inhibiting now your coagulation cascade. So this slide just tells us or shows us the importance of your vitamin K and its cycling. So as you can see here, no, glutamate residue here is present in the vitamin K dependent clotting factor. You have your 9, 10, 7, and 8. Okay? For this glutamate residue to be converted into carboxyglutamate, di ba yung carboxyglutamate, sila yung may dalawang COO negatively charged, no? So for it to be converted into a carboxyglutamate residue, it would need the active form of your vitamin K, which is your vitamin K hydroquinone. Vitamin K hydroquinone, once already utilized no, in the carboxylation of your glutamate residue would then be converted into your vitamin K epoxide. This epoxide form is inactive form of vitamin K, no? So hindi siya nagagamit for the carboxylation reaction. Okay? So how do we do or how do we recycle epoxide to be converted into hydroquinone? Kasi nga yung hydroquinone yung kailangan natin. So we have an enzyme known as your reductase enzyme, no? So to convert your vitamin K hydroquinone or vitamin K epoxide back to its hydroquinone form. So pag nabalik na siya dun sa hydroquinone form niya, it can again be used for the hydroxy or in the carboxylation reaction. Okay? Okay. Let me just share my screen again. Okay, so this is just another picture no showing that same concept no. So they have vitamin K that is utilized by your vitamin K dependent carboxylase is in the hydroquinone form. Okay, once your hydroquinone form is consumed, no, you need to convert it back into its. It will be converted into epoxide form, and in the process of reduction, no, um, this vitamin K would then be converted again to hydroquinone form to be used again by the same enzyme. So na-discuss naman na natin yan. Same here. Ito lang yon. So how does warfarin inhibit your vitamin K? Di ba nga sabi natin kanina, yung kailangan natin for the carboxylation nung clotting factor proteins is the hydroquinone form. This hydroquinone form is utilized by the vitamin K dependent carboxylase to be converted again to its epoxide form. Yung epoxide form, no, hindi siya nagagamit tong enzyme na to. So it would need to be converted back again to hydroquinone form. And we have enzyme for that. It would undergo two steps reduction no, to be converted back here. Warfarin no, would inhibit the enzyme no, the reductase enzyme. So kapag inhibit niya yung enzyme, babalik pa ba si vitamin K epoxide to its hydroquinone form? Hindi na. May magagamit pa ba si clotting factor or vitamin K dependent clotting factor para ma-activate yung 9, 10, 7, and 2? Wala na. So warfarin in that sense can be used as an anticoagulant because it would inhibit no the reformation of vitamin K hydroquinone. Okay, so this is just a slide no, or a table showing you the differences between heparin and warfarin. No, so heparin is a large acidic polymer. Okay, while your warfarin is a small lipid soluble molecule, warfarin can only be taken orally. Heparin is given parenterally. Onset of action of heparin is within seconds, while warfarin you need to take it for a few days, no, for it to act, no? The mechanism of your heparin would be activation of your antithrombin 3. So naalala nyo kanina, it potentiates the activity of this thrombin inhibitor, while your warfarin would inhibit no your vitamin K no via the inhibition of the reductase enzyme. Antidote for heparin toxicity would be your protamine, no? Protamine. Sorry, I would like to correct myself. Protamine is a positively charged molecule, no? So once it associates with heparin, no, it would activate this negatively charged molecule. While your warfarin, no, there's no uh, there is no actual antagonist of your warfarin, no. So you either supplement no the patient with the hydroquinone form of vitamin K, or you give the patient fresh frozen plasma to supply them with the clotting factors that they need. So heparin no, is used for a short period of time, while your warfarin is usually used for weeks to even months, no, for anticoagulation. Okay, so let's now go to the last portion would be your fibrinolysis. So we've already discussed about platelet aggregation. We've discussed about formation of your fibrin. And after our body is done, no, um, repairing the damaged vessel, no, we need to remove the fibrin or the clot, no, to the process of your fibrinolysis. This is carried out by your plasmin, no, your plasminogen to plasmin. And this plasmin would then cleave no your fibrin, no? They will dissolve the fibrin clots to form your fibrin degradation products. Okay, so what are the components of your fibrinolysis? You have your plasminogen, which is an inactive zymogen of your plasmin that binds to fibrin. So doon siya nagbabind sa mismong fibrin. So may incorporate siya dun sa clot to be converted into your plasmin. Okay, which is actually the active form responsible for degrading your fibrinogen to your fibrin degradation products. So you need to remember, no, as long as they are bounded to fibrin, they are in their active form. Okay, and we have an inhibitor of your plasmin in the form of your alpha 2 antiplasmin. So if you inhibit plasmin, no, if you inhibit plasmin here, there will be no molecule to convert your fibrinogen to fibrin degradation product. So how does your plasmin convert or degrade your um, clot? No, so tissue plasminogen activators, no, as well as your urokinase that is found in most of the tissues in our body would cleave, no, the arginine and valine bond, no, in your plasminogen. Kapag na kapag na-cleave na yung arginine and valine bond no, in your plasminogen, it would convert now your plasminogen to plasmin. And this plasmin now is capable of doing no its action. So ano bang ginagawa nung plasmin, no? So plasmin would specifically bind to lysine residue. Naalala nyo kanina yung lysine and glutamyl residue na pinag-bind natin kanina? So this plasmin residue would bind to lysine residue, no, to be incorporated into the fibrin mesh and would cleave this, no. And I apologize for the technical problems that we're experiencing. So again, let's just emphasize, no, yung plasminogen to plasmin, no, would be activated by cleavage of arginine valine bonds, no. Kapag ka-activated na siya, yung plasmin, may incorporate siya dun sa lysine residue and would cleave now your fibrin. Next would be your inhibitor, no, of your fibrinolysis. We have this your activated thrombin activatable fibrinolysis inhibitor or your TAFI-A, no. So your TAFI-A do is it removes your terminal lysine residues from the fibrin. Kapag tinanggal mo yung lysine residue here, meron pa bang pagbabindan yung plasmin? Wala na. Madedegrade pa ba niya yung fibrin? Hindi na. So in this sense, your TAFI-A would inhibit your fibrinolysis. Okay, so tatanggalin niya yung lysine residue. So this TAFI-A is activated by your thrombin as well. So next, you have your tissue plasminogen activator. So they are serine proteases that are released in the circulation after vascular injury. No, sometimes we can also synthesize it, no, and be given to the patient to a patient, no. So they are inactive unless they are bound to fibrin. So they form soluble degradation products and dissolves the clot, no. So paano nila dinidissolve yung clot? So ina-activate nila yung plasminogen to plasmin. No? So yung plasmin will be the one to dissolve the clot. So urokinase, also an example of activator of your plasminogen. So it is originally isolated from the urine. O kaya siya tinawag na urokinase. So its primary mechanism of action is to degradation of the extracellular matrix. So this is just a summary of your fibrinolysis. Your plasminogen would be activated by your tissue plasminogen activator and your urokinase, no, by cleaving your what do you call this? By cleaving the bond between your arginine and your valine. So in that sense, it would be converted into your plasmin. Your plasmin would then be incorporated in your fibrin, no, through the lysine residue, forming now your fibrin degradation products, no. You have also your plasminogen activator that is capable of, plasminogen activator inhibitor that is capable of inhibiting your TPA. And you also have your TAFI-A that is capable of inhibiting your TPA as well as the incorporation of your plasmin to your fibrin. Paano nga niya uli in-inhibit yung incorporation? By removing the lysine residue. Okay? So these are just different molecules that are synthesized by the endothelial cells now that would have roles in the process of thrombosis and fibrinolysis. You have your ADPase that is actually formed no in the dense granules of your platelet uh, that is capable of degrading your ADP to AMP. So itong enzyme na to, sorry, I would just like to correct myself. This enzyme would degrade the ADP that is secreted in the dense granules to AMP and PI. So in that sense, would that inhibit the platelet aggregation? Yes, kasi ano nga uli ang action ng ADP? This will promote platelet aggregation. You have an enzyme that degrades this. So that would inhibit platelet aggregation. Next would be your nitric oxide. So nitric oxide would inhibit platelet adhesion and aggregation by increasing the level of your cyclic guanosine monophosphate. You also have your prostacyclin, no, which is also inhibitory to platelets by increasing the levels of your cyclic AMP. Next would be your thrombomodulin. No? So when it binds to your thrombin, as you can see a while ago, no, it cleaves your protein C to activate it. Protein C together with your protein S would degrade your clotting factor 5A and 8A. So it actually inhibits coagulation. No? So para maalala, it modulates your thrombin. Next would be your endothelial protein C receptor. So it facilitates protein C activation by the thrombin thrombomodulin complex. And lastly, we have your tissue plasminogen activator, which activate your plasminogen to plasmin, no, by cleaving your arginine and valine bond, no. So this plasmin activated plasmin is capable of degrading now your fibrin. Okay, I think that ends my lecture, or that ends the part 2 of our lecture, and I would like to thank you guys for listening.